In this presentation, we will focus primarily on the reproductive cycles of both males and females. We will review the anatomy briefly. You should understand the anatomical relationships and terminology before delving into reproductive physiology. You can do this by reviewing the material from Biology 250 or doing the anatomy review found in Interactive Physiology available on Mastering A and P.
The testes make gametes and secrete male sex hormones. Production of sperm, and targets for hypophyseal hormones, are found in and between the seminiferous tubules that comprise the testes. Let's take a closer look at an enlarged portion of the seminiferous tubule epithelium. The first cells that are encountered are just inside the connective tissue capsule surrounding the seminiferous tubule. These cells are diploid or 2 in spermatogonia. The spermatogonia are the stem cells for all the sperm a male will make throughout his lifetime. As we move toward the lumen of the tubule, we see spermatocytes in various stages of meiotic division and maturation. The lavender-colored sustentacular cells support the developing gametes both physically and physiologically. They also form a blood-testis barrier which protects the spermatocytes from attack by immune cells and maintains the appropriate chemical environment for optimal sperm production. We will see later that the sustentacular cells also secrete hormones that help to regulate testosterone levels within the seminiferous tubule. There is a lot of overlap between the endocrine system and almost every organ of the body, and the reproductive system is no exception. Reproductive function in males is regulated hormonally, and there exists feedback loops between the hypothalamus, pituitary gland and the testes. This relationship is called the brain-testicular axis, which takes about 3 years to get regulated. The function of the brain-testicular axis is to balance male hormones to ensure that enough sperm are being produced, and to regulate secretions of the glands and maintenance of male secondary sex characteristics. When the sperm count falls below 20 million/ml of ejaculate, the male is essentially sterile. Conversely, overly high levels of testosterone are harmful for normal spermatogenesis and other functions. Here's the short version of how optimal levels of testosterone are maintained both in the bloodstream and within the seminiferous tubule. The cells within the testes that are involved are the interstitial cells and the sustentacular cells. Under command of the hypothalamus pituitary gland, the interstitial cells secrete testosterone. The testosterone enters the bloodstream via the capillaries located outside the seminiferous tubules. This testosterone maintains target tissues associated with being male. Testosterone is also needed inside the seminiferous tubule to support sperm production, however the sustentacular cells form a blood-testis barrier. Therefore, the sustentacular cells produce and secrete androgen-binding protein which combines with testosterone and pulls it into the seminiferous tubule. When testosterone levels within the seminiferous tubules get too high, the sustentacular cells secrete a hormone called inhibin which stops the release of hormones from the hypothalamus pituitary complex so that there is not too much testosterone within the testes. When blood levels of testosterone get too high, it feeds back to the hypothalamus pituitary gland which slows down the release of hormones that cause its formation. Note, FSH and LH are the same hormones in both males and females. In males, the testes are the target organ, the ovary is the target in females. LH in males is also called ICSH for interstitial cell stimulating hormone, since the target of LH, ICSH, is the interstitial cells. FSH causes the sustentacular cells to produce androgen-binding protein. One of the functions of testosterone is to support the production of sperm. Spermatogenesis is the process of making mature haploid, N, sperm for the diploid spermatogonia. The process is divided into 3 steps. Spermatocytogenesis involves mitotic division of the spermatogonia. Recall that the spermatogonia are the source of all the sperm a male will make. Therefore, the spermatogonia must make identical copies of themselves via mitosis. When a spermatogonium divides, one of the 2 daughter cells stays behind to remain as a spermatogonium and the other begins its development toward becoming a mature sperm. The daughter spermatogonium will then undergo 2 successive cell divisions resulting in the formation of 4 haploid spermatids from a single spermatogonium. This process of reduction division is called meiosis. During meiosis the number of chromosomes is reduced to half, from 46 to 23. Each diploid cell of our body has 46 chromosomes or 23 pairs, one set of the pair F from the mother and one set from the father. Fertilization and formation of a zygote restores the diploid number. If there were no meiosis, then each successive generation of offspring would double their chromosome number, 46 plus 46 equals 92, 92 plus 92 equals 184, etc. Variations in the chromosome number of a species leads to either death of the embryo or severe genetic disorders. The next step of spermatogenesis is called spermiogenesis. This process involves an extreme makeover and reorganization of the spermatid cytoplasm to form a mature, mobile sperm cell. This picture illustrates the dramatic changes that take place in structure of the spermatids during the process of spermiogenesis. The Golgi apparatus produces hydrolytic enzymes necessary to break down the oocyte's membrane and forms the acrosomal vesicle. Meanwhile, cytoplasm is being shed and reorganized. The nucleus condenses and the acrosomal cap moves to the region that will become the head of the sperm. The mitochondria reorganize and form a spiral around the base of the tail. The tail is a flagellum and the mitochondria supply the energy that propels the tail. Note the acrosomal cap will be the part of the sperm that contacts the oocyte first. The nucleus is greatly condensed and there is very little cytoplasm. The mitochondria form a spiral around the midpiece, which will power the tail, which consists of a flagellum. The table above summarizes the source, stimulus and major effects of testosterone. The targets of testosterone include certain neurons in the brain, as well as accessory reproductive organs, ducts, glands, and the penis, causing them to grow and assume adult size and function. Development of the male secondary sex characteristics is also due predominantly to testosterone, which begins exerting its effects at puberty. These include the appearance of pubic, axillary, and facial hair, enhanced hair growth on the chest, deepening of the voice as the larynx enlarges, the skin becomes thicker and more oily, the bones increase in size and density, and the epiphyseal plates close in late puberty. Testosterone also raises basal metabolic rate and is the basis for sex drive, libido. The male reproductive system is anatomically more complex than that of the female, but the female reproductive system is physiologically much more complex compared to males. The role of the female reproductive system not only includes production of the female gametes, oocytes ova, but her body must also nurture and protect a developing fetus during 9 months of pregnancy, and continue to nurture the baby by producing milk in the mammary glands. In this presentation, we will study the anatomy of the female reproductive organs, describe the reproductive cycles of the ovaries and uterus, and what happens if the reproductive system is successful in producing an embryo. Once again, refer back to the presentation on the anatomy of the female reproductive system or to your textbook. Some of the anatomical information will be presented briefly in this presentation. We will discuss the features of each of the organs comprising the female reproductive system individually, but keep in mind that all of them function together for one purpose, propagation of the species. We will begin with the primary sex organs, the ovaries. The ovaries are paired, almond-shaped organs that are only about 5 cm in diameter, smaller than the testes. They are homologous with the testes in origin and in development. The surface of the ovary is covered by a simple cuboidal epithelium known as the germinal epithelium. This is a very misleading term because it doesn't give rise to anything, and the term germinal implies that something develops from it. In the old days it was thought that it was the source of the female gametes, but now we know that this is not true. Just inside the germinal epithelium is a capsule of dense CT called the tunica albuginea, as it is in the male testis. The inside of the ovary contains the stroma and developing ovarian follicles. The tear part of the stroma is called the cortex. This is where the ovarian follicles are found. The inner stroma is called the medulla, and this is where blood vessels enter and leave the ovary. Embedded in the cortex are follicles which contain oocytes in various stages of development. The oocytes are surrounded by follicular cells. At any one time, in the ovary of a woman in her reproductive years, various developmental stages of follicles containing oocytes can be found. An ovarian follicle consists of an oocyte surrounded by cells referred to as either follicular or granulosa cells depending on the stage of development. The most immature and least developed are the primordial follicles. They form clusters in the outer cortex called egg nests. These follicles are the reservists. They are dormant and not growing under the influence of pituitary hormones. Primary follicles are beginning to grow under the influence of FSH or follicle-stimulating hormone from the pituitary, and Th. Follicular cells begin secreting estrogen. As development continues, some will grow larger, develop multiple layers of cells called granulosa cells which begin to secrete a fluid called the liquor folliculi. One of these will become the follicle of the month and it will grow larger and larger as the cavity inside the follicle called the antrum continues to fill with fluid and stretches the surface of the ovary. Some of the granulosa cells form a stalk that supports the oocyte called the cumulus oophorus. A mid-cycle surge of luteinizing hormone, LH, causes ovulation, and the oocyte with a ring of granulosa cells attached called the corona radiata is expelled into the pelvic cavity. What is left behind of the follicle becomes a separate little endocrine organ called the corpus luteum. It secretes estrogen, but mainly progesterone. We will see that progesterone acts on the uterine lining in such a way as to support gestation, gesterone. The corpus luteum has a limited span. If fertilization does not occur within 14 days of its formation, it involutes and dies, forming a white scar on the surface of the ovary called the corpus albicans. Formation of the corpus luteum, yellow body, takes place approximately in the middle of the menstrual cycle, day 14 of a 28-day cycle. Following ovulation, which results from the LH surge from the pituitary gland, the corpus luteum becomes a separate endocrine gland within the ovary. The granulosa cells remaining in ovary and the cells of the theca lutein, interna, a cup-like structure formed from stroma surrounding the follicle, form the temporary endocrine gland in a process called luteinization. The granulosa lutein cells secrete progesterone, and theca lutein, interna, cells produce estrogen. These two hormones target the lining of the uterus, endometrium, preparing it for implantation of an embryo should fertilization take place. Oogenesis is the process of making a secondary oocyte and takes years to complete. During fetal development, two in oogonia multiply rapidly by mitosis and then enter a growth phase and lay in nutrient reserves. Gradually, primordial follicles begin to appear as the oogonia are transformed into primary oocytes and become surrounded by a single layer of flattened follicular cells. The primary oocytes begin their first meiotic division, but become stalled late in a stage called prophase I, and do not complete it. These follicles remain in suspended animation all through childhood, about 10 to 14 years. At puberty, about 250,000 oocytes remain and a small number of primary oocytes become activated each month by the mid-cycle surge of LH. The one that will eventually be ovulated, continues meiosis I and produces two haploid cells each with 23 chromosomes. The smaller cell is called the first polar body. The larger cell, which contains nearly all the cytoplasm of the primary oocyte, is the secondary oocyte. The secondary oocyte will be ovulated, but it won't finish its reduction division, known as meiosis II, unless it is penetrated by a sperm cell. With that, the second polar body is expelled, leaving a mature ovum with a single copy of the haploid number, 23, of chromosomes. The process of oogenesis is very different than spermatogenesis. One spermatogonium divides twice meiotically, producing 4 mature haploid sperm, and this process is continuous. In oogenesis, only a single mature ovum is formed from the oogonium, and this happens only if the secondary oocyte is fertilized. The female reproductive cycle consists of the coordinated events occurring in the ovary, ovarian cycle, and the uterus, uterine or menstrual cycle. It is a tightly regulated process involving hormones produced by the hypothalamus, adenohypophysis, ovary, and corpus luteum. The primary function of the female reproductive cycle is to get an oocyte ready for fertilization, and to get the uterus ready to receive a zygote should fertilization take place. The ovary is the target of the pituitary gland, adenohypophysis, and the uterus is the target of the ovarian hormones and corpus luteum. The ovarian and uterine cycles occur in non-pregnant females between the ages of 11 to 50. They involve cyclic histological and physiological changes in both the uterus and ovary. Each cycle takes 1 month. Above is a schematic of the hormonal regulation of the ovarian cycle. On day 1 of the cycle, rising levels of GnRH from the hypothalamus stimulate increased production and release of follicle-stimulating hormone, FSH, and luteinizing hormone, LH. FSH exerts its main effects on the follicular cells of about 20 or so follicles. LH targets the thecal cells. As the follicles grow, LH causes the thecal cells to produce androgens. These diffuse through the basement membrane, where they are converted to estrogens by the granulosa cells. The rising estrogen levels in the plasma exert negative feedback on the adenohypophysis, inhibiting release of FSH and LH. Within the ovary, the increased release of estrogen causes the granulosa cells to release inhibin, which also exerts negative feedback control on FSH release. Inhibin prevents more than one follicle from developing to maturity per month, which limits the possibility of producing multiple embryos. Once circulating levels of estrogen reach a critical concentration in the blood, it exerts positive feedback on the hypothalamus and pituitary. This results in the LH surge from the anterior pituitary occurring about mid-cycle. The LH surge stimulates the primary oocyte of the dominant follicle to complete the first meiotic division, forming a secondary oocyte that continues on to metaphase 2. LH also triggers ovulation and the formation of the corpus luteum, luteinization. The newly formed endocrine gland begins producing progesterone and estrogen. Rising levels of these two hormones exert negative feedback on the release of LH and FSH. The corpus luteum also releases inhibin, which prevents another LH surge that could cause a second ovulation. The estrogen and progesterone secreted by the corpus luteum targets the glands of the uterine endometrium, preparing them for an embryo should fertilization take place. If fertilization does not take place, the corpus luteum degenerates, and the blood levels of estrogen and progesterone sharply drop. This stops the negative feedback on FSH and LH secretion, and the cycle starts over. This diagram details the events explained on the previous diagrammatic representation of the ovarian cycle. It also shows the histology of the structures in the ovary that are responsible for the hormonal regulation. The ovarian cycle is divided into two consecutive phases. The follicular phase is the period where the follicles are growing under the influence mainly of FSH. This phase typically lasts from the 1st to the 14th day of the cycle. The luteal phase is the period when the corpus luteum is active, days 14 to 28. This diagram shows the hormonal events occurring during the ovarian cycle as a series of graphs, which makes it easy to visualize the relative timing of hormone release from the hypothalamus, pituitary gland, and levels of circulating hormones released by ovarian structures. The top two graphs show the pulse frequency of gonadotropin releasing hormone from the hypothalamus and the response of the pituitary tropic hormones. Figure C shows the stages of follicle development as a result of FSH and LH, and graph D shows the circulating levels of the ovarian hormones estrogen, progesterone and inhibin. Note that if fertilization occurs, the corpus luteum is kept alive. In fact, part of the chorion, extra-embryonic membrane surrounding the young embryo, will become the fetal contribution to the placenta. It is the source of human chorionic gonadotropin, HCG. The embryonic HCG keeps the corpus luteum alive so that it continues to secrete estrogen and progesterone. This maintains the endometrium so it and the developing embryo are not shed. HCG can be detected in the bloodstream as early as 10 days after fertilization, peaks at 2 months, then declines. The maternal contribution to the placenta is modified endometrial cells called decidua because it will be shed after birth, named after the shedding of leaves from deciduous trees in the fall. The placenta is completely developed by the 3rd month of gestation. At 2 months, the placenta is big enough and takes over the secretion of estrogen and progesterone. Therefore, the placenta, not the corpus luteum, maintains the endometrium. Throughout the remainder of the pregnancy, placental progesterone and estrogen also suppress ovulation so that there is no possibility of another zygote being formed. The placenta also secretes human placental lactogen, which prepares the mammary glands for lactation following birth. The uterus is where a young embryo will implant if fertilization takes place. Interestingly, the uterine endometrium is only able to support implantation of an embryo for a very short period each month. Therefore, development and preparation of the endometrium must be precisely timed to support the embryo, which begins implantation 6 to 7 days after ovulation. To this end, the changes in the endometrium are coordinated with the phases of the ovarian cycle, which are in turn dictated by gonadotropins released by the anterior pituitary. Before beginning how ovarian hormones make structural and functional changes in the uterus, Let's review briefly the layers of tissue comprising the uterus. The wall of the uterus consists of 3 layers. The outermost is the perimetrium or sericea. The middle layer is the myometrium, which consists of 3 layers, an inner longitudinal, middle circular layer which is the thickest of the 3, and an outer longitudinal layer. The innermost layer is the endometrium. The endometrium is the primary target for hormones produced by the ovarian follicles. It is lined with a ciliated, secretory epithelium, and contains uterine glands. It is the superficial part of the endometrium, called the functional layer, that is affected by the ovarian hormones and is sloughed off during each menstrual cycle if fertilization does not occur. The basal layer stays behind as a source for rebuilding the endometrium during the next reproductive cycle. There are numerous B lymphocytes and plasma cells in the lamina propria to prevent infections that could spread to the peritoneal cavity. The uterine or menstrual cycle is divided into 3 phases. The first phase is called menses and corresponds to the time when the functional layer of the endometrium detaches from the basal layer. The next phase occurs between 7 to 14 days, and is called the proliferative phase because the functional layer of the endometrium is beginning to grow back as a result of ovarian estrogen. Notice that menses and the proliferative phase are occurring during the follicular phase of the ovarian cycle. The third phase, occurring between day 15 to 28, is called the secretory phase. This corresponds to the luteal phase of the ovarian cycle. Ovulation has occurred, and the corpus luteum is producing estrogen and progesterone. The endometrial glands begin secreting glycogen into the uterine cavity. These nutrients sustain the embryo until it has implanted in the blood-rich endometrial lining. Estrogen induces the appearance of secondary sex characteristics of females, which includes the growth of the breasts. Recall that development of the mammary glands begins during the 7th week of embryonic life as a thickening of the epithelium that runs from the axilla to the inguinal region. This region is called the milk line. Typically, in humans, only 2 regions along the milk line develop into mammary glands. But supernumerary glands may form anywhere along the milk line in both males and females, but usually only the nipple develops and appears more like a mole. Mammary glands are modified apocrine sweat glands, consisting of 15 to 20 lobes. Each lobe is divided into lobules which are made up of the secretory alveoli. Surrounding each secretory alveolus are myoepithelial cells which move milk through ducts towards the nipples. Estrogen stimulates the growth and maturation of the breasts at puberty and maintains their adult size and function. Cyclic changes in estrogen affect the growth of the epithelial cells in the ducts and alveoli. During pregnancy, the mammary glands are exposed to estrogen, progesterone and human placental lactogen. Prepare the glands for milk production by causing the secretory alveoli and ducts to grow. Suckling of the nipples results in sensory information carried to the hypothalamus. Causes the release of prolactin from adenohypophysis and oxytocin from pars nervosa. Prolactin stimulates milk formation. Oxytocin causes milk ejection, milk letdown reflex, by causing contraction of myoepithelial cells and glands resultant. G in forceful ejection of milk from the nipple. The table above summarizes the source, stimulus, effects and targets of the 2 primary female sex hormones, estrogens and progesterone. As in the male, estrogen and progesterone have many targets in both body and brain. These hormones underlie the development of the female gametes, as well as control growth, metabolism, sexual behavior, pregnancy, and female secondary sex characteristics.